I work in the Division of Genomic Medicine at NHGRI, so I am obviously someone who believes that there is some promise to genomic medicine. And so what I wanted to do today was talk a little bit about what we can actually do today in the clinic and some of the places where genomic medicine is already being used, and then also talk a little bit about some of the things that are coming for the future, some of the things that have been making headlines, and where we think this could potentially go and what some of the challenges are going to be in getting us there. So I wanted to start off with just discussing a little bit about what is genomic medicine. So you may have heard a number of different words bandied about in this area. People talk about genomic medicine and personalized medicine and precision medicine, and what is the distinction between all of these different topics? So personalized or precision medicine really refer to the same thing. It's just people's individual preference over whether they would rather talk about it being personalized or precision. But both of them are really talking about using medicine tailored to an individual and a person that they're seeing in the clinic. Now, this isn't really new or novel. That's the way that medicine has been practiced always. The medicine really is about treating an individual rather than something like public health, which is about treating populations. But personalized medicine starts really trying to focus this in on getting more information about the patient that's in front of you and trying to use that to tailor the care and therapy. And you'll often hear three specific areas talked about when people mention personalized or precision medicine. The first of these is really integrating lifestyle or behavioral choices. So these are things that someone has control over. You can decide how much are you going to exercise, what are the foods that you're going to eat, are you going to smoke or not? Those are all things that you're choosing about your lifestyle. They also typically talk about including information about your genes, so your genetic information and how that might influence your individual risk of getting a disease, or potentially how that information might help you with responding to a specific treatment. So could we pick a specific intervention for you that's based on your individual genomic profile? And then the third item is really the environment. So this is all the things that are happening then outside you. So where do you live? What is the environment that you're exposed to on a daily basis? And trying to incorporate all of these different items into an individual's care is what characterizes this idea of personalized or precision medicine. Genomic medicine is then a specific type of precision or personalized medicine, where you're talking about using that genetic genomic information in care. So when people talk about genomic medicine, it really could be a type of personalized or precision medicine, but they're typically then focusing on the genomic aspects of the care and decision-making process. This also is not something that is new to medicine. Doctors have been using genomic information in care for many years. We're just used to talking about things like family history. So when you go into your doctor, one of the things they typically ask you is, does your family have a history of cancer or cardiac disease? Have you had heart attacks in your family? That is a form of genomic medicine. They're getting information about your family and whether or not you are potentially at an increased risk of developing any of those conditions based on the fact that you might have inherited genes from your parents that put you at increased risk of having that condition like cancer. So this really is, once again, building off of something that's already being done in medicine today, but trying to do it in a little bit more specific or detailed manner. There are lots of different technologies that can potentially then be used in genomic medicine, everywhere from family history through doing tests for individual genes to doing biochemical assays that are trying to see the actual genetic effects. Are you actually seeing the outcomes from having a variant, to doing sequencing to try and map the DNA in your genome. And what I'm going to focus on today is talking about sequencing and some of the different ways that it's being used in genomic medicine. So my next section is just to talk a little bit about what is genomic sequencing. So DNA sequencing basically gives us the order of the bases that make up our DNA. And so there are 4 different bases that code all of our DNA, A, C's, G's, and T's. They pair together so that A's always go with T's and C's always go with G's. And these base pairs then can get read across the DNA molecule. And what sequencers are doing is reading that code in lots of different ways. And basically all of the advances that have been made in sequencing technology are trying to find ways to be able to read these bases more accurately, quicker, and in ways that require smaller amounts of samples for a lower cost price. And so we've gone from the Human Genome Project, which mapped the first human genome back in 2003 and cost billions of dollars, to today you can get a genome sequence around the order of about $1,000. So the cost of this has come down dramatically. And what we do with that sequence information is try and determine which parts of your code have genes, and sometimes also looking for the regulatory instructions that tell those genes then to turn on or off. Because when you have changes in these regions, it can then potentially lead to disease phenotypes. And one of the things that I wanted to point out here, because it matters a little bit for some of the technology, is that within each of these genes, there are basically 2 different kinds of code. We talk about these as being introns or exons. The introns are basically code that gets cut out, and the exons are the part that is actually coding what's going to end up making the proteins, which are what control the different functions in your body. So that's the part that's going to actually get translated into a product. And that's important because there are 2 main types of sequencing that get talked about a lot today in the news. One of these is genome sequencing. And in genome sequencing, you're looking at the entire genome, the whole pie, everything that you can find, which is over 3 billion base pairs in humans. So it's a lot of data and information. We still can't get all of it. So even when people talk about whole genome sequencing, we really aren't getting every single one of those base pairs, because every technology that gets used has some things that they miss. There's always going to be some amount of error in the reads, and then there's always some technologies have preferences for things that they read better than others. So there's always going to be some segments that they just have a harder time being able to get to. So genome sequencing gets us most of that whole pie. Exome sequencing, on the other hand, selectively picks out those exons, those coding regions of the genome. By doing that, you can sequence less of the actual base pairs, so you're able to then get more coverage of each of those individual reads. And this allows it to be a little bit cheaper than doing genome sequencing. And this picks out about 1% of those 3 billion base pairs is what's going to be making up those exons, or those coding regions. And once again, this is one of those places where it's about 1%, this small slice of the pie, but it also is going to be spread over the entire genome that you're actually picking those out. So once we have the sequence, we need to know what makes potentially these changes or variations that are going to lead to disease. And one of the most common types of changes that gets looked for are these single nucleotide polymorphisms, or SNPs. And basically what these are just a change in a single one of those base pair codes. And so in this example, you can see there's data from 4 different individuals. And in that yellow region, you can see that sometimes people have that TA, sometimes they have the CG. Versus in all the regions before that, the code is always the same between individuals. So that just lets you then find regions that are different between individuals. And there are also other types of genetic variation that you can pick up by sequencing. You can find insertions, which is basically when you put in a single base or sometimes more, so you're adding in a section of code. You can get deletions, where you're taking out a small region of code or more than one base. And another one that gets talked about a lot in the news are copy number variation. So this is where you have a segment of your DNA that gets replicated a certain number of times. And different individuals have different numbers of copies. So in this example, some individuals have 3 copies, some people have different variations in copies, 5 and 3 or 3 and 4. So the number of those copies changes. And this can also play into some disease phenotypes. So once we have these different variants, the idea behind genomic medicine is that if you have a group of different people, that they're going to have some difference in the genetic variants that they exhibit, and that if you knew what that information was, you might be able to pick different treatments for them. And that's the general idea behind how genomic medicine typically gets played in public advertising, or if a medical center is talking about how they want to be able to use genomic medicine for clinical care. So now that we've talked a little bit about what genomic medicine is, I wanted to give a couple of examples about what we can do with sequencing today. So 3 areas where genomic medicine gets used today are within diagnosis, screening, and therapy. Starting off with diagnosis. One of the main areas where sequencing has been implemented is with rare diseases. And in the United States, we have an official definition that rare diseases are conditions where there's less than 1 in 200,000 people in the United States that has that condition. And that makes them very rare and therefore difficult for a doctor to be able to diagnose when they have one of these individuals come into their clinic. Because these are the kinds of people that you're not going to be seeing every day. They're going to be presenting with different kinds of signs and symptoms. And it can be very difficult with some of these rare diseases to be able to determine which of the over 7,000 different conditions that we already know exist an individual might have. So lots of options and limited amounts of data that a clinician is going to have initially. So based on that, it can be difficult for rare disease patients to be able to get a diagnosis. And a survey that was done recently showed that these individuals are typically going more than 7 years before they're able to reach a diagnosis in the United States. And typically, in order to get to the point of that diagnosis, they've gone to 8 different doctors before they get to the point of someone being able to tell them what it is that they have. And sometimes there's even been a couple of other diagnoses that they got along the way that were misdiagnoses. So either someone thought they had something and then they got another test and it turns out that wasn't what they had, or sometimes these individuals who have really rare conditions will get labeled with a diagnosis even if the clinician knows that it's wrong, because they need to call them something in order to be able to get them a treatment or a therapy, or to be able to bill for the services. And because of that, it's really difficult to be able to get these individuals to the point of knowing what their condition actually is. So in this rare disease space, you might have a patient that goes to their first clinician and they do a clinical evaluation and they're not able to determine what the individual has, so they send them to another doctor who runs a series of tests and gets back some results and does another evaluation, and maybe they have some ideas, so they refer the patient on to a specialist who then does another clinical evaluation and runs their own series of tests. And this just continues going on for that period of about 7 years, where people are going to multiple different doctors and sometimes having new tests run and sometimes having the same test run, and it's just not a very efficient process. So the idea of where genomic medicine might fit into this is that you could do an exome or a genome sequence earlier in the process, combined with that clinical evaluation where you're learning about the patient and their phenotype and other testing, and that this might help you get to a diagnosis for these individuals at an earlier point in time. So does this work? So some of the earliest examples of this have been looking at patients that have been undiagnosed, which typically means that they're those patients who have been on that really long diagnostic odyssey. They've spent years, they haven't been able to get a diagnosis, and doctors aren't quite sure what to do. And that's been where a lot of the tests have been done so far, where exome or genome sequencing has been applied in a clinical setting. And in those settings, exome or genome sequencing has been able to make a diagnosis in about 25 to 35% of these cases who have gone years without being able to get a diagnosis. So that still is a significant number of patients who are getting an answer to something that they've gone years not having an answer to. In general, pediatric or younger patients tend to have a slightly higher rate of diagnoses. We seem to be able to make a diagnosis in those patients a little bit easier than in adult patients. We're not quite certain why that is, but some of the ideas that have come up is that it might be due to the fact that when you have pediatric patients, you have the family available. So you have the child as well as their parents, and being able to have those parents available for testing as well can help you with being able to find a genetic variant that might be affecting the child. Because you can look and see if this is a condition that's recessive, where you have to have two copies, one that you inherit from your mother, one that you inherit from your father. You can see, do you see that in the parents that aligns with what you're seeing in the child? You also can look and see if you see something that's truly new in the child that the parents don't have. That might make you think that that's also might be more likely to be associated with the condition. And we talk about these as being de novo variants that have occurred newly within the child. And so having that parental information, it seems like doing sequencing of these trios, so the family, the parents, and the child, helps with making more diagnoses. There's also some evidence that it might be due to the fact that adults have other factors that influence why they have disease as well. They have all of those lifestyle and environmental factors that we had talked about earlier. And because they've lived a longer life, they have more of that plays into their health. So it could also be that part of the reason why it's easier to be able to make a genetic diagnosis in a younger individual is because it's more likely that their disease has more of a genetic component to it, versus the adult may have some of these more lifestyle or environmental factors that are also influencing their disease. So this is just one example from the Undiagnosed Diseases Network, which is a program that I work with at the NIH. And within this program, we accept individuals that have gone through a long diagnostic odyssey and been unable to reach a diagnosis through standard of clinical care. And this is one patient who was seen by one of the clinical sites in the Undiagnosed Diseases Network. He presented to the UDN at 20 months of age, so very young, with seizures, cataracts. He had microcephaly, which just basically means a small head size, and global developmental delay, so was having trouble with developmental milestones and being able to reach those. And by doing sequencing in this individual, they were able to come up with a new variant. But it's just one individual with one new variant that's never been associated with another human that has that same disease or condition. So what do you do then? What a lot of science has been really moving towards now is the idea of trying to share the information as much as possible within these communities, to be able to try and find other patients that might have the same conditions, and that by sharing a lot more of this data and information that we're now being able to collect, this idea of big data, that you might be able to then help find more information and more cases that are similar and be able to make more diagnoses. So we took the information about this one patient that was seen as part of the Undiagnosed Diseases Network and put it into a database that allows for clinicians as well as patients themselves to enter information about their condition and a genetic variant that they have, and see if there's anyone else who matches in that database. And through that, we were able to find another 6 patients through different variations of that matching database, through some other clinical labs, and through some just putting information out on the web with these participant pages where we said, we have a patient who has this condition. Do you have someone who looks like this? And it would be found if you just Googled that gene, you could find this page. And using all of those methods, because there were then 7 patients who now all had the same variant and similar types of phenotypes, they were then able to publish a paper then saying that this variant is associated with this particular set of symptoms developmental delay, epilepsy, cataracts, and define a new syndrome related to this NACC1 gene. And then once that's published and this information gets back into the databases, it then can be used to be able to help others who would just be going through a standard clinical exome or genome screening. People can then know that that variant is associated with this new disease.
Is it cheaper to screen if you already have a pretty good idea of what you're looking for?
So if you know what you're looking for, then you can do just a single gene test, which is cheaper than looking at the entire exome or genome at this point in time. So that does help some with being able to then follow up on testing as well. So how does this matter? So this is a different case from another one of the projects that I work on, and this is Sebastiana, who is the infant that you see in the photograph here. And when she was born, she started having seizures. They were able to control them by using a combination of different seizure medications. But the problem was they were making her really drowsy. She was having problems with being able to feed. And if they started reducing the amount of medication that she was getting, then she would start seizing again. Sebastiana was very lucky in the fact that she was born at a hospital where she had access to a group that is doing really rapid sequencing. And this rapid sequencing protocol allowed them to be able to get sequencing results back within a matter of days rather than weeks, which is kind of the current clinical standard. And by using that information, they were actually able to identify which gene it was that was causing her seizures, and then start her on new medication, which actually controlled the seizures, but then didn't give her all of the drowsy effects because it was the right medication to be able to treat her seizures and the actual condition that she had. And so this is just one example of how on the extremes, at very young ages with very rapid sequencing, you can have some pretty dramatic impacts even with the way that sequencing is done today. This is being done clinically in areas. As a rapid protocol, there are actually some clinical labs that will now do this as a rapid screen. Doing it in a matter of days is typically difficult because it requires basically everyone to be able to stop what they're doing and run everything through on a nonstop kind of protocol. But being able to get things back within a matter of a week or two is something that can actually be ordered as a clinical test from a number of different laboratories at this point in time. So it's really changing the way that we can use this type of sequencing information to be able to make diagnoses in a more acute setting where the patient really does need an answer soon because they're sick in like a critical care state and need to be able to have an answer to be able to affect their care. Yes?
As I understand it, what you were saying was you had the sequence of the child's genome, but is it that you didn't know which exome might be producing the observed effects, which is why you had to go out to the broader population and kind of back into it, retrofit it, say, okay, all these 6 patients have this identical exome, so this must be what produces the manifestations.
Yes, so in this example here, when you have a new variant that hasn't been associated with disease before, everyone has variations in their genome. So any person's going to have a couple of, variants that are out there that look like they might potentially be disease-causing. When you find a new variant in a child that's sick, you're looking to see, does that gene seem to make sense biologically? So do you think that that could potentially be related to the condition? You might have some data in the literature about the function of that gene. What does it do? But if you don't have any information about other cases or that this has been seen or associated with the disease, when you only have one case, you're still really not sure, is that the right one? So having multiple cases that all then have that same variant and the same phenotype then gives you more evidence that it's that variant that's causing the disease. That's not just a spurious finding.
Do you need 2 or 5 or 10?
More than
Yeah, exactly. So it's a real question out there right now. So in general, to be able to publish, you need at least 2 cases to be able to prove to a journal editor that it's likely that your finding is real, that you didn't just happen upon this by chance. Because the reality is there is a small chance that you could just happen to find a variant in a gene that you could find a plausible biological story for, but it's not actually what's causing the disease. In general, the more the better. People are always happier when you have more of them. But with these really ultra rare conditions, the reality is there might not be more people out there. And so that's part of, trying to figure out with this kind of genomic medicine, how can we combine the data as much as possible between groups of hospitals within the United States, globally, to be able to find those other cases? And then are there ways that you can follow up on this using other methods, like doing functional studies in model organisms, to be able to get other data or information? Because you might have the only case in some really, really rare conditions.
I mean, part of the fun is being able to figure out what these altered genes actually do. In this example, what did they discover the NACC1 gene actually did?
So they are still working on all of the detailed mechanisms. And so this is a case where, they have some evidence from, functional studies, but that moreover, what they have is 7 cases that all have the same phenotype and the exact same change in their DNA. So a lot of times it's one of those where it can actually be quicker to find more individuals who have the same variant than it can be to be able to do some of the functional studies in model systems, because those studies can take months or years to be able to actually understand then the actual mechanism of how that variant is causing your disease. I wanted to also talk a little bit about where some of the different misunderstandings come then, where these different types of stories get talked about in the news. So one of the misunderstandings that comes out of these kinds of diagnostic stories is that this kind of sequencing really only applies to rare diseases, and that genomic medicine is only going to benefit rare diseases. And one thing to note related to that is that while these rare diseases are all really rare by themselves, there are lots and lots and lots of them. So we know of probably about 7,000 or so. So that's about 30 million people in the United States, which is 1 in 10 Americans. So when you start talking about lots of very rare things, they end up actually being pretty common in the population as far as an impact. It's just that each one of them is different, and that makes it really hard to be able to study them. So this actually does have a pretty big impact on individuals. And then as we go through some of these other examples, we'll talk about some ways that sequencing and genomic medicine can also be applied in some more common conditions. So the next topic I wanted to talk about was screening. So screening is when you're taking a set of individuals that don't have any symptoms. So in the case before, we're talking about sick individuals that you're trying to diagnose. Here you're talking about apparently healthy individuals and trying to learn something about what their risk might be. So in this particular example, I wanted to talk a little bit about carrier screening, because it's one of the places where sequencing is now starting to be used more, but that's been used in medicine for a while. So in carrier screening, you're taking 2 parents who are both apparently healthy individuals, and you're looking to see if they are carriers for conditions that they might pass on to their children. Typically, these are recessive conditions where you need to get 1 variant from your mother and 1 variant for your father. And if you happen to get both of those variants that are abnormal, then you end up with the condition. So if you have your parents are both carriers, about 1/4 of their children would be healthy, about a half of them would be apparently healthy but also carriers, and then you'd have about 1/4 of them that had a chance of having a condition. And so carrier screening is used before pregnancy to be able to determine, do you have a risk of having a child that might have one of these conditions that's more common within the population. And originally, a lot of carrier testing was based on ancestry. So if you're of Ashkenazi Jewish descent, then you would get tested for Tay-Sachs and cystic fibrosis. And if you're African American, you might get tested to see if you have sickle cell disease. And it was looking to see, do people have these variants that are associated with very specific populations? But what people have been realizing, and what's being recommended more today, is that it's getting harder and harder within the United States to be able to tell what someone's ethnicity is. There's more mixing of different populations, and just because these variants might be more common in one background and one ancestry doesn't mean that someone from a different ethnic group might not get that condition. It just means they have less of a risk of potentially getting it. And so what's been happening now more in carrier screening is to do these pan-ethnic screening, which is the idea that rather than picking which diseases we're going to test you for based on your ancestry or your ethnicity, we're going to give the same set of condition testing to everyone, regardless of what their ethnicity is, in order to be able to see if they have any of these conditions.
Is that common in pregnancy now, that all women who are pregnant are getting these screenings?
So the current recommendations from ACOG, which is the American College of Obstetrics and Gynecology, which kind of governs these things, is that they would like to have carrier screening offered to all couples that are intending pregnancy. So that it should be offered to everyone. It is considered to be optional, so people can say we don't want to do that. But the current medical recommendation is that everyone should be offered this if they are considering a pregnancy.
Is it expensive?
So there are different types of testing that can be done, and depending upon how many conditions you're testing for, the cost can also vary. So yeah, right now there's still variation within the community as to what different clinics are going to be offering. The current guidance is that a clinic should offer the same standard test to everyone. And then if someone asks for a specific panel, they should talk to them about the risks or benefits of that panel as opposed to what they normally would use for testing. And then if they still choose that, they should be able to offer and provide that to them if that's what they want.
Okay. But they're doing the basic sequencing on both parents recommended for everyone.
So there's different ways that it can be done. So sequencing is the newest way that you can do this. Many of the tests that are being done today are still panels where there are specific genes and variants that are being tested for, and you're only testing for that specific gene or variant. But there are now ways that sequencing is also being used, because especially when you have lots of different populations, the variants that are typically on these panels were all detected based on the ancestry or ethnicity of the populations that were most likely to have that condition. So they're the most common variants, but that doesn't mean that there aren't other rare forms that might still cause that same disease. So sequencing is a way that you can potentially find some of those other rarer variants in populations that might not have normally been associated with a condition.
But the cost of doing screening for every pregnant couple is just huge.
And this is one of those where, then it starts becoming, well, how detailed do you get, and what is the benefit of then being able to do that type of screening? And so there are a lot of studies that are now trying to look at where's that right balance between catching more people and being able to provide them with the information about these conditions and making sure that everyone in the United States, regardless of what their ethnic background is, has a chance to learn this information. It's not just based on who happened to have had the most sequencing or genetic testing done in the past, and balancing that versus the cost of doing the testing and the access factor of if the test is very expensive, people might not all be able to do it. So cystic fibrosis is actually a condition that is then now been recommended both by the American College of Obstetricians and Gynecologists as well as the American College of Medical Genetics and Genomics. So these are two professional societies that govern OBGYN and genomic medicine, clinical genomics, to be screened for everyone. And so here you can see an image which talks about the prevalence rates of cystic fibrosis in different types of populations. In general, cystic fibrosis is still more frequent in Caucasian backgrounds, but it can be found in these other ethnicities. And what sequencing has found is that sometimes it's just that there are also different variants that are found in some of these other ethnicities versus some of the really common ones that had been previously detected. But overall, across all of these populations in the United States, 1 in 31 Americans is a carrier for a variant in the CFTR gene that's associated with cystic fibrosis. So it's pretty frequent within the population that someone might be a carrier. And so that's why there's been this recommendation that this is one of the few conditions that multiple professional societies have recommended screening for.
But that doesn't, now you don't infer that 1 in 900 would then have CF, because you might have, they might be different variants that the two parents would have, and so they're both recessive, and so neither of them is expressed.
So there's lots of different variants. Not all of the variants actually cause disease either, and there are variant combinations that have stronger or weaker effects. So there's a lot of nuance beyond that fact that just people are a carrier. And this is one of those conditions where, as they've learned more about it, it started off with one variant that was particularly common in Caucasian populations, and then they found more variants, and by finding some of those other variants, then that helped lead to being able to develop some more therapies. And sometimes you can then go from a therapy that might be very specific to a specific rare variant, but it teaches you about the biology of the condition that then allows you to develop things that work for broader populations. So why is this kind of screening important? And one of the major reasons why these professional societies recommend doing carrier screening is so that if couples find out that they are both carriers for the same condition and have a risk of having a pregnancy that might have that condition, that they can get genetic counseling. And the idea is that they get to talk to a genetic counselor who's trained to be able to talk to individuals about the risks associated with having a child with that condition. So what is the likelihood that they're going to have a child with that condition? They can also talk to them about the condition itself. What is it like to have a child with that condition? And then some of the different options that they might be interested in pursuing based on the fact that they have carrier status. So if an individual is a carrier, they can talk to individuals about getting donor gametes. So this would be getting a sperm donor or an egg donor, so that you knew that you didn't have two copies of that variant that were going in to being able to make the child. You can also talk about pre-implantation genetic diagnosis. So this is where, if they're doing IVF, they can actually today take a sample from the embryo cells that go on to develop the placenta and do testing of those specific cells and determine whether or not it's likely that that embryo would end up developing one of these conditions. So you can actually select for IVF for selecting embryos that don't have conditions. You can also do prenatal diagnosis. So you can do testing in the prenatal period to be able to determine, is the child going to be born with one of these conditions, so you can do planning. And a lot of times that has to do with trying to make sure that if there's potentially risks to the pregnancy, that the mother's aware of them, that you can plan for being able to have a hospital birth if that's going to be important, that the care team is ready, that you have the resources available to be able to deal with the pregnancy going forward. And so those are all some of the options that carrier screening allows for a couple to then be able to discuss with a genetic counselor.
What are donor gametes?
So that's just getting, so gametes are your sperm or your egg cell. And so you could get from either like a sperm bank or from egg donors, other gametes that you could use so that you weren't using both parents. Exactly. And so within the screening, one of the common misunderstandings that comes up is linking screening to diagnosis. And screening really isn't the same thing as diagnosis. And they can get really confusing when people are talking about these in a clinical setting, especially when people are talking about using sequencing technologies for doing both. So in a screening test, you're taking individuals who are apparently healthy and trying to determine if they have a risk for disease or potentially very early disease that you wouldn't have noticed because they seem to be healthy individuals. That then has to be followed up with a diagnostic test, which is then done in an individual where you are trying to determine, do they have disease or not? And so that's then a more definitive test to be able to determine, do you have a specific diagnosis? Do you have this condition or not? And it's important that these two get considered separately, because typically when you're doing a screening test, you want to make sure you catch anybody who might potentially be at risk. So these tests are designed so that they might have lots of false positives that come up, because you don't want to have a false negative. You don't want someone to get missed who was really, truly at risk because of the test. But then when you're doing diagnostic testing, you want to make sure that person actually really does have disease. So the way that the tests are designed is a little bit different because of that. And then finally, I want to talk just a little bit about how this is being used in therapy. So one of the areas where genomic medicine has been used the most in therapy to date is within cancer genomics. So cancers are genetic and have genetic factors that influence them. And the genome of a tumor, so sequencing the actual tumor itself, can then help with treatment decisions to be able to target your therapy to that tumor to be able to get rid of the cancer. And there are two ways that trials are now being designed today to be able to try and take advantage of this. Because one of the things that we've learned about cancer is that even though there's many different types of cancers that may originate in different parts of the body, that they may have some underlying genetic similarities. So in the first of these types of trials, the one with the people that are in lots of different colors, you have a number of individuals. All of those different colors are all different places in the body that their cancer originated. So some of them might have breast cancer versus lung cancer versus colon cancer. But if you look across all of these individuals, the different colored dots, if they're red or white, indicate that some of these individuals all share a genetic factor that was causing them to have a risk for that cancer that is shared, a driver for that cancer, that's shared across the different types of cancers. And so in these types of, they're called basket clinical trials, you're taking a whole bunch of different people and then you're lumping them together into a basket based on the fact that they have a genetic variant shared amongst them, regardless of where their cancer originated. If they have colon cancer, breast cancer, lung cancer, they have a genetic change that is the same, so that you can try and see if you can use a therapy that's targeted specifically to that genetic change. In the other example here is an example of an umbrella trial, which is another new clinical trial design, where in this case, all of the individuals are the same color because they all have the same kind of cancer. So here we could say that these individuals all have colorectal cancer, but they have lots of different colored dots. So they have lots of different kinds of genetic variants that are driving why they have that cancer. And so here, within that same area, origin, they all came from the same place, you're splitting these people up into different therapies, once again, based on those different genetic variants, and therefore testing different drugs to see if they can affect these individuals. So it's a way of trying to bring this idea of genomic medicine to cancer therapy. And an example of groups that are doing this is the NCI, the National Cancer Institute's MATCH and pediatric MATCH trials. And these are a combination of both this basket umbrella design. So they take individuals across multiple different types of cancers, and then they sort them out by doing some genetic testing, see what variants they have, and then they try and match those to a set of different experimental therapeutics that they have available. So they have a set of drugs that target certain mutations, and then if individuals end up having those variants, they can get assigned to those different clinical trials. And so what they're trying to do is find out if you were assigning people to a therapy based more on their genetic factors rather than the type of cancer that they had, can you get a response? And these are some of the early results from just earlier this year that came out from the MATCH trial. And what you can see is that in one case, there were 24 out of the 65 patients that had variants in a particular gene that ended up with over 6 months of progression-free survival. So their tumors didn't end up shrinking. There wasn't a change in the cancer itself, but they stopped progressing and getting worse. So that stable state is still something that people look for in cancer treatments. But these individuals had different kinds of cancer. So some of them had lung cancer, some of them had bile duct cancer. So it wasn't just where it had started, it was related more to giving them a therapy related to that gene variant that they had. And in a second case, the drug that they were giving was related to these HER2 overexpressing tumors. And in 3 of the patients that had really rare cancer types, they actually saw over a 30% shrinkage of the tumor in those really rare cancer types. But overall, almost 50%, so 46% of the patients, once again, had this progression-free survival. And once again, it was over a variety of different cancer types, so ovarian, uterine, and colorectal cancer. So this lends the idea that you can get potentially some better treatment matches looking at the genetic causes of these cancers and tumors versus just classifying people based on the site of origin of their cancer. Do you have breast cancer or lung cancer? And though it might not seem, as exciting that these people are not, getting a reduction in their tumor size in most of these cases, it's that they're being held stable. The reality is that in order to even be considered for this trial, over 30% of the individuals that were in these two clinical trial arms had already received over 3 different lines of cancer therapy. So they've already gone through 3 different sets of what standard of clinical care and practice is with no results. But when they got this therapy that was targeted to their specific variant, they did see a stabilization of their cancer. And so one of the things that gets discussed a lot here when we talk about therapy and using genomic medicine is the idea that a treatment success is equal to a cure. And so a lot of times in the media, people will talk about, we got this result and that was great, but patients really want cures. And unfortunately, where we're really at right now is that a lot of times in these clinical trials, it's similar to what we just talked about in the match, where you might be getting a halt to progression. You might be getting a delay in the onset of symptoms. This is still good because this is still beneficial clinically to the patients. But just because something is working and providing evidence that doing more of this genomic medicine type of pairings might be beneficial, it doesn't necessarily mean that this is directly going to lead to cures for these diseases.
But the phrase curing the condition, would that entail reorganizing the part of the genome that was causing the condition? I mean, what would curing the condition, what does that mean?
Yeah, typically if you're talking about trying to get to cure, you're getting to the point where someone isn't needing therapy anymore and they no longer are then exhibiting any signs or symptoms of disease. And there are potentially ways that, you could give something that would get you to the point where you've really cured the disease, where that person no longer has that condition. But a lot of these therapies today are really more about trying to be able to stop or halt progression of symptoms versus being able to really go in and alter the genome and trying to make it so that it never happens. They're still typically treatments rather than.
But these ones that you heard, for example, as anchored to this exome or part of the genome, curing would entail turning the effect of that off, right, at that level.
Yes, so there are trials that are looking at doing gene editing, where the idea is that you are actually changing something either in the cells in an adult's body now so that they are actually changed and then would produce the normal form of the protein or what you need to be able to get rid of the condition. And that is something that's being talked about. But so far, there's been very few trials that are doing that kind of editing. Most of this is about trying to target a therapy to a biological pathway where we're then trying to be able to prevent symptoms from developing. Mm-hmm. Yep. Okay, so then things for tomorrow. So last couple of minutes, I want to just highlight a couple of things about topics that had come up a lot when we talk about genomic medicine and where we are today with them and where we might be able to get in the future. And one of these is this idea of lifetime genomic medicine, and the other is the idea of tailored therapeutics. So looking at lifetime genomic medicine, this is a concept that gets talked about a lot when people talk about the future promise of genomic medicine. The idea that at some point in time in the future, everyone could get sequenced at birth and then would have this information available for the rest of their lifetime for their medical care. And that this would help with improving their health, it would help with determining what they're at risk for, and it would help with being able to tell what drugs you would need to be able to give someone, because if someone got sick, you'd already have that genetic information available in their medical record to know what drug they might respond to best. And though this is something that is technically possible, it is still really an idea that's in the future from the standpoint that there's a lot of information that's still needed to really make this work. So though we can get an exome or a genome and we can put it in your medical record, in order to be able to really interpret it and understand what all those variants mean, you need lots of longitudinal data over individuals and how those variants have been affecting them over entire lifetimes. And building that knowledge base is going to take time to be able to understand what all those variants do, so that when you have the genome, you know how to interpret all of those different changes. And then the second piece is that you also need the infrastructure to be able to then access that information and maintain it. So as we're developing that knowledge base, you need ways to be able to actually look at the information again and update it with new knowledge. And you need ways to be able to access that data when a clinician needs it. They need to be able to actually get that information back out of the medical record in a way that's user-friendly, quick, easy to understand for what is an entire genome's worth of data. So you can't expect a clinician to know what every one of those base pair changes is going to do. We need the computational infrastructure to be able to help make this kind of genomic medicine possible. So while it's something that is possible, it really is still a future implementation that people are working on, developing that knowledge base and the infrastructure to make it work.
But from what you were saying earlier about the pregnancy screening, it sounds like it's now standard practice to be doing the sequencing. So the data is starting to be accumulated for every birth.
So there is sequencing that's being done in the carrier screening, in prenatal testing, in some cases looking at newborns. So there is sequencing data that's being collected. The issue is that right now there's still not a lot of standardization about how that information gets stored in electronic health records. All of these hospitals need to have electronic health records, but there's not really standards about how they have to be able to communicate between one another. If you go from one doctor to another, how do you get your information from one to the other? And how do you access that? Because a lot of times what's coming back still is a report that's in like a PDF file. So it's basically the electronic equivalent of someone putting a paper document in the record. You can't really access that very well. You can't use it for something else in the future. It really just gives you a document that a doctor could read now and be able to use immediately. It doesn't have that usefulness for the future component. And then the other example I wanted to give was with tailored therapeutics. And so this is a very recent example. This is Mila, and her story was just announced in October of this year, where they were able to do a single individual clinical trial of a customized drug therapy that was created especially for Mila, based on the genetic variant that she had for Batten disease that was unique to her. They were able to develop this drug in a year time frame from the time that they identified this variant through doing some animal model testing, getting a manufacturer to develop the drug, going through the FDA process of approving, being able to give it, and actually providing it to Mila for treatment. And from initial reports of the first couple of months worth of treatment, Mila has Batten disease, which causes you to go blind and then start having seizures and neurological problems, and it is universally fatal. And she was at the point where her seizures were starting to get more and more frequent, which is how they were able to get a compassionate use case approval from the FDA to be able to do this trial in a single individual. And from the start of the drug so far, it seems that it has reduced the numbers of seizures that she's having in a day, as well as their frequency. They went from being seizures that could be minutes long to seconds long, which is a major improvement for her as far as quality of life. But as amazing as this story is, and a true credit to the fact that this mother and her family and all of the researchers that worked on the development process, it really is a case of a single individual. And this type of story shows kind of the promise of where genomic medicine could go. But at this point, this really was a trial that was very expensive. the parents themselves raised millions of dollars to be able to provide the funding to be able to do this research, which is just something that not everyone has the time or access or ability to be able to do. And it also was really a case of all the stars aligning. This was a case where it was a very unique variant that was found that was related to another drug for another condition that had just been tested and designed by the FDA, where they could try using a similar process. And it was a single individual who had a potentially fatal disease where they were progressing rapidly. Therefore, the FDA was willing to be able to give the approval to be able to do the trial. But what this really does show is that this was based off of another rare conditions finding, where it was then able to be applied to a new individual who also had a rare disease. And what the rare disease community is really working on today is trying to find ways to make it so that those kinds of drug repurposing and learning from one another and creating things that might be able to be used for multiple different rare conditions, how do we build that infrastructure so that this can happen for more cases without this having to be a case where all the stars aligned and someone was able to get that miracle cure. And so with that, I have just a couple of conclusions from today. We've talked a little bit about how DNA sequencing technology can be used in multiple settings for diagnosis, screening, and therapy, and some of the different misconceptions around those uses. And then we've talked a little bit about some of the possibilities for tomorrow with lifetime genomic medicine and tailored therapeutics. I think the main message I would want to send people away with today is that a lot of this just takes time. That genomic medicine does have a lot of promise, but when you hear some of these really great stories in the news, they typically are the cherry-picked top of the amazing stories where things all worked out and aligned. And though this is being used in the clinic, you can get exomes and genomes ordered from a clinician today to do diagnosis. You can use it for doing screening today. In order to be able to make this really a practical reality for everyone and used as part of the standard of practice is still going to take time to be able to get the research to make that happen. And I hope this has been interesting and would happy to take any questions from everyone.
Just out of curiosity, how easy is it to switch off a gene or switch on a gene? Just curious as how.
So it's going to depend on your gene because all genes have different ways that they're being turned on or off. So you have different regulatory regions. In general, there are technologies that can actually, you can cut out an entire gene and then you're not producing it at all. But if you want to be able to actually control that regulatory machinery, then it really is a case of you have to be able to understand that element that's controlling the gene. And there's different types of those elements, and some of them are controlled by different factors. So you might be able to have something that worked for multiple different conditions or multiple different genes, but how easy that is going to be able to turn on or off is going to be really dependent on that mechanism, unfortunately. Yeah.
I have two questions. One is, why is the cost of doing exome screening versus whole genome screening not, you said it was difference between less than 1,000 and more than 1,000, but it's 100-fold difference in the amount of information. Why is the difference? Why isn't whole exome screening far more expensive? And also, the cost. It seems to me that this lifetime genomic medicine is something that maybe only the wealthy are going to be able to afford. I can't imagine insurance companies covering this for everybody. So we might have a sort of a technology that's only available to the wealthy.
Yeah, so with regards to the cost, even though you're getting the whole genome versus the exome area there, the reality is you're really not doing very much more sequencing. It actually takes more work to be able to get an exome than it does to be able to get a genome. Because when you want to do an exome, you have to do a step to be able to select for just those exons. So in the laboratory, you have to be able to do an additional step to be able to select out in a library just those individual exons that you want to be sequencing together. And then you're sequencing just those exons. But typically, what you're doing is you're sequencing them multiple times so that you get higher levels of coverage. So you might be doing the same amount of sequencing as the genome, but in the genome you might get 10x coverage, and in the exome you might get 30 or 40x coverage. Now, I mean, sorry, the other way, you might get 100x coverage in your exome, and in the genome you might get like 10x kind of coverage. So because of that, it still takes a lot of sequencing. You're still using a lot of sequencing when you're doing an exome. And the savings is really on, maybe you can fit more individuals on a single chip, depending upon what your technology is. You might be able to do a few more exomes on a run, and that's where you're getting your savings. But you're still typically doing a lot of sequencing, and you have to do that extra step. And then with regards to the cost and insurance piece, so this is something that we do a lot of talking about at NHGRI, because there is a real desire to try and make it so that genomic medicine doesn't become something that's really just the rich benefit from this, but where it can actually become a part of clinical care. There have been some arguments made that for insurance companies, especially for some of these conditions where people were going for years of odyssey, and they were doing lots of different testing and lots of different care that wasn't having any impact, that doing a sequence earlier on, even though it might be a more expensive test to start with than whatever the first test was they were planning on doing, that switching to that earlier in the process could save the insurance companies a lot of money of doing testing that wasn't getting you to a diagnosis. And so far, it seems like, there are insurance companies that are interested in learning more about this and ways that it could potentially be applied more broadly in clinical medicine. Within the Division of Genomic Medicine at NHGRI, a lot of what we do with our research programs is trying to figure out how to close those gaps. So a lot of what we're doing is trying to figure out how do we best spend our research dollars to be able to make it so that there is a healthcare system where these electronic medical records can talk to one another, so that clinicians can use this genomic medicine information in clinical care. So what are the problems that need to be solved regarding that infrastructure? What tools need to be developed? What can we do regarding standards and coming up with ways that those can be developed to be able to make the information easier to be able to pass between different groups? And what can we do with regards to some of the technologies to be able to push those towards being more accessible and developing the knowledge bases to hold that information? So we've actually been working on developing a database that's related to ClinGen and ClinVar. What they basically do is it takes all of the information from the clinical labs that are doing this kind of sequencing testing, and originally they all just had their own databases, and everyone had their own variants that they had seen, and they would look against their own database and they would say, have I seen this, yes or no? What this gives them is some place to put that data. And so they now can put that information in this one place, and now they can look across all the labs that are doing clinical sequencing and be able to see if someone else has that variant, which just helps moving that kind of information exchange forward. Yeah.
Does that imply you're going to be involved in big data in the future?
So we do some work with big data and infrastructure and computational programs. It's a much bigger problem than just within genomic medicine, but it is definitely something that the NIH is doing a lot of work on, trying to figure out how to utilize those technologies to be able to help with health applications.
These ancestry websites now where you send in a sample, and are they doing sequencing? What data are they accumulating? Because it sounds like they're developing repositories.
Yes, so most of the ancestry direct-to-consumer testing companies that are online, where you can order, a saliva sample kit, you send it in, and they give you back results, are doing genotyping versus sequencing. Typically, so that means they have specific variants that they are looking for in the genome that they test for. And then they use that information to be able to determine what your ancestry is. Typically, some of them are what we refer to as being ancestry informative markers, meaning that they are either unique to certain populations, or we understand the differences between different populations and the frequency of those variants. And then that allows them to be able to help classify you into different ancestry groups based on the information that we have about those populations. Some groups will also provide back some health information, and that's typically because they're directly genotyping some of these variants where there are known health information available that they are then providing back as well. But typically, part of the way that testing works is that you are basically consenting when you send in samples for them to then have access to that information. And you are getting access to be able to view it and utilize the tools that they've developed for being able to understand the information that they're providing back to you. But in return, they then also have access to the data for being able to then do their own research, and some of them do therapeutics or other types of development as well with that information within their own database.
So could a pregnant couple just send their saliva to one of these things and they get the result and then they give that to their obstetrician?
So it happens, yes. So people will, send samples out, get information back, and then come in to their clinician with a report, who then has to figure out, what was the test that you had done. These tests are typically not going to be the same level as what you would have if you were getting clinical testing from your doctor, and then try and figure out, is there something there that they can interpret? But typically the issue here is that the reports that are coming back are designed to be read by consumers, a participant who's getting just some general information about this, and are not friendly for, a clinician to try and read through, a 12-page document in 15 minutes and then know how to be able to use that for your care. So it's once again that information exchange problem of getting lots of data at once that doesn't necessarily have context to then use for care. Right. Some of these companies do share some of the information from what they find into some of these other databases. So though they have their own individual databases, some of them are sharing some of this information and doing other research studies. And if they get money from any NIH source to do a research study, they have to share those findings back with some of these publicly accessible databases. There are also databases where an actual participant patient themselves can take the results from a genetic test that you had and put them into databases yourself and share that information and decide how much information you want to provide with that and put it into, for example, the matchmaking that I had talked about with the Undiagnosed Diseases Network. That matchmaking database, the Matchmaker Exchange, has a couple of places that feed in patient information where you can put the information in yourself and access that resource. So there are some places where that's being built, but typically they still are framed where some resources are geared more towards the clinicians or researchers and some are geared more towards a participant kind of experience.